Welcome back. In this section, we're going to learn all about erythrocytes. So, an erythrocyte is a red blood cell. Cyte means cell, erythro means red, so red blood cells. So, these are pictures of the small one is an individual red blood cell, and on the right is a scanning electron micrograph of blood, and it's been artificially colored. So, the red ones are the red blood cells. If we were to take a sample of your blood and look at a cubic millimeter of it, so if you took a tube of blood, which is a centimeter, and you took a tenth of that, which would be a millimeter, and you made a little tiny cube, that would be a cubic millimeter. In that cubic millimeter, there would be 5 million red blood cells, 5 million erythrocytes in that small amount of blood. Okay. When we look at your red blood cells, you can see that they're not round. They actually have dimples, and the dimples go in on both sides. So we say their shape is biconcave. Cave means it's going in. So concave goes in, and bi means on both sides. So this biconcave shape makes the red blood cell much more flexible as it's cruising around in your bloodstream. And it also gives it a greater surface area than it would if it was just, a rectangular cell, which means it's got more surface area for picking up oxygen or dropping off gases as well. Now, the red blood cell, the reason why it has this biconcave shape is because it's missing many of its organelles. Okay. So if there was a nucleus there, you would have that bulge there in the middle. There's no nucleus, which means there's no DNA, which means there's no transcription or translation that can happen. So there's no DNA replication, which means the cell cannot reproduce, which means the cell is dying. In addition, there's lots of other organelles missing, including no mitochondria. So there's only glycolysis that can happen, right? We can't have the Krebs cycle. We can't have the electron transport chain. So we can only make 2 ATPs at a time. So let's hope it doesn't need a whole lot of ATP. So that means if we've gotten rid of a lot of organelles, we must need this space for another purpose. What is that purpose? This red blood cell is 90% full of something called hemoglobin. So you ask, well, what is hemoglobin? Well, let me show you. Here's a hemoglobin molecule. As you can see, it's quite complicated. What I want you to realize, first of all, there are 4 globin chains. This picture I like because it shows that there's 2 different types. So it's used pinkish purple for one type and browns for this other type. So there's 2 of each of the 2 types of chains. You don't have to know the different types of chains. As adults, we have 2 different types of chains. We call them alpha chains and beta chains, but you don't need to know that. When we are in utero before we're born, one of these types of chains is replaced by a fetal type. Okay, but here's our normal adult hemoglobin. See this thing that looks kind of like a spaceship? There's one of these attached to each of the globin chains, and this is known as a heme molecule. So here's what a heme molecule looks like if you were to draw it out in its chemical form. And what is important to realize is in the middle of this heme molecule, there is an iron molecule, Fe, that is iron in the center of the heme. And this is important because it is to this iron, which is a positively charged molecule, that oxygen, which is negatively charged, will bind. Okay, so this shows that one oxygen can bind to each of these irons. Okay, so basically what we're saying is this erythrocyte has become a bag of hemoglobin to carry oxygen throughout your body. So when the erythrocyte travels into your lungs, oxygen is going to diffuse through the simple squamous epithelium in your lungs and attach to all the iron on the heme molecules attached to the globins of the hemoglobin. Then it will be carried around in your bloodstream until it reaches the tissues where it'll drop off the oxygen. Now, this picture shows that the hemoglobin molecules, when they arrive in your lungs, have no oxygen, and then they travel in your lungs with lots of oxygen. And then when they get to your tissues, they're releasing all the oxygen, but that is not actually true. In reality, I said each hemoglobin has 4 globins, so that there's 4 hemes, so that there's 4 irons, so that there's 4 molecules of oxygen. So in reality, what happens is when it goes to your tissues, usually only one of those molecules of oxygen is dropped off and the other 3 are still there. Okay, which means if you quit breathing, it can circulate a whole other time and drop off another oxygen as well. Okay. So this brings me to a question I need to ask you. How much oxygen do we carry in each individual red blood cell? I'll let you have a minute to calculate it. How many you came up with? 4? Yeah, it's not 4. Mm-mm. Do you think we would create an entire red blood cell 90% full of hemoglobin to carry 4 oxygen molecules? Mm-mm. That would not be efficient. The problem is, I told you your red blood cell is 90% chock full of hemoglobin, but I didn't tell you how much hemoglobin there was in a red blood cell. You just jumped to 4, but that's how much iron was in one hemoglobin, and you assumed 1 hemoglobin per red blood cell, when in fact, each red blood cell contains 250 million hemoglobin molecules. So now calculate how much oxygen is in a red blood cell. Yes, multiply 250 million times 4. No, you don't have to draw all the zeros. Just do 250 times 4, then add 6 zeros to it. Or do 25 times 4 and add 7 zeros. So every red blood cell, yes, why do you multiply by 4? Because every hemoglobin attaches 4 oxygen molecules. So now you know that every red blood cell can carry over 1 billion oxygen molecules. Yes, that's the correct answer. All right. Now, in addition to carrying oxygen, blood also carries carbon dioxide. But carbon dioxide's a little bit more complicated. Okay. It gets carried 3 different ways. So make sure you understand the 3 different ways and kind of understand the proportions. So here is carbon dioxide being given off as a waste product in cellular respiration. Yes, remember that electron transfer chain where you were generating carbon dioxide and also the Krebs cycle? Okay, so here's that carbon dioxide. It's going to get dumped into your venous blood where 7% of it is going to stay dissolved as carbon dioxide. And you're going, Dr. Powell, why only 7%? And I'm going, okay, so what does dissolved carbon dioxide in a liquid give you? Well, carbonation. That's fantastic if you have a soda. But carbonated blood, bubbles in your blood, not so good, because that means you can't carry oxygen in the bubbles, right? You can't have red blood cells in those bubbles. All right. So 7% of it is dissolved in the blood, which means we got 93% of it, which is not going to be dissolved. All right, so now 23%, so almost a quarter of it is actually going to enter the red blood cell and bind to hemoglobin. Okay, so no, it does not bind to the iron. It has nothing to do binding to the heme. It binds someplace totally different. And whether or not there's oxygen bound has nothing to do with whether or not carbon dioxide can bound. Okay. So that, once again, that's still a minority of it. So we still have 70% left over. So what's the other 70% going to do? Well, it's going to go into a chemical equation where eventually it's going to end up back in the plasma as this. Oh, so this is an equation that you must learn. So yes, it's going to be on test 4 and it's going to be on test 5. So 70% of your carbon dioxide is going to be carried as this HCO3 with a minus sign, which is called the bicarbonate ion. It's going to be carried in plasma. So there is an enzyme at this arrow and an enzyme at this arrow. It's the same enzyme which quickly makes this reaction happen. And here's what's really cool. It's the same enzyme that carries the reaction this way and carries the reaction this way. And this is what is keeping your blood pH constant. Because remember, this hydrogen ion is what's affecting your pH. Remember? pH, the H of pH has to do with hydrogen ion concentration. So you need to know not only the chemical symbol equation, but you also need to know the word equation. So CO2 or carbon dioxide is going, because that's what we're throwing into the blood, is going to bind with water. Why? Because our blood is plasma is 90% water. All right, so we got water. Okay, so now cover up the middle thing and just add up how many C's, how many O's, how many H's. It doesn't matter what order you put them to because this is not a chemistry class. If you were to put them in the correct order, it would be H2CO3. But if you write it CO3H2, I will give you credit for this class. Doesn't matter to me. Just make sure you have the correct number of C's, O's, and H's from when you add carbon dioxide and water together. This substance, when carbon dioxide and water are added together, is called carbonic acid. It is an acid, which means it wants to give away a hydrogen ion. So yes, this is going to break down quickly into a hydrogen ion, which is an H, and you must put the plus sign, as well as a bicarbonate ion. So now we've taken a hydrogen away. So if you take one hydrogen away, what you're left with is HCO3, and you must put the minus sign for the bicarbonate ion. So learn this equation today. And remember, you're just starting with water and CO2. Come up with your middle carbonic acid, take away your hydrogen ion, subtract that, and what you're left with is going to be your bicarbonate ion. And this is our major buffer system in our blood. Okay. So let's talk about the life cycle of our erythrocyte. As we learned before, when we learned about the process of hematopoiesis, we learned that all of our cells of our blood were made in our bone marrow. So we have stem cells in our red bone marrow that are going to make our red blood cells. And so instead of calling it hematopoiesis, this would be erythropoiesis, formation of red blood cells. And then the cells are going to circulate in your blood for about 120 days, 4 months. While they're circulating around, it's just kind of like your backpack while you're walking around on campus for a semester, for a month, it gets kind of dinged up. And remember, when you were a child and you had a backpack over, at school at the end of the year, you would get a new one. You would throw out the old one and you would get a new one. Well, after 128 days, that erythrocyte's really banged up and it's going to be destroyed in the spleen. Now, we don't throw it in the trash, but we recycle the components. So here's what's going to happen. The globin, part of the hemoglobin, is going, because it's a protein, it's going to be broken down into amino acids for protein production. Heme, the iron, is going to be taken off and either stored in the liver, or if it's needed right away, going to be reused in the bone marrow. The remaining heme minus the iron it's going to be broken down. So if you've ever had like a big fall or a big injury and had this big bruise, like right away it was red and then it was blackish and you could see it turn green and yellow over time. This is showing the heme being broken down. And so let me show you actually in a picture what this looks like. But this is all you need to know. Then what I'm going to show you next has more details which you don't need to know. Okay, so we start over here with this erythropoiesis in the red bone marrow. Here's our red blood cell traveling around for 128 days. It's all dinged up. Now it needs to be broken down. Macrophages are going to do phagocytosis of it. The globin is broken down to amino acids and can be reused for any kind of protein synthesis. The heme molecule gets broken down. The iron gets taken into the bloodstream, and the blood has to have a carrier protein that is carried back to the liver where it can be stored, or if it's needed, a carrier protein will then take it back to the bone marrow. And then the rest of the heme molecule gets broken down into something that gives you like a green bruise, and then the yellowish bruise, which is taken into the liver and secreted. And that greenish stuff, if you've ever seen a greenish or yellowish when you vomit, that's bilirubin. And the bilirubin actually gets excreted from your liver into your small intestine, where it gets further broken down in your intestines. And this is what makes your feces look brown. And part of it gets reabsorbed and sent to your kidney. And this is kind of what gives your urine its yellowish color. Okay. So globin broken down for proteins, iron travels in the blood, stored or reused. The heme broken down leaves the liver and the bile, and eventually broken down to feces and urine. All right. So let me talk about something called erythropoietin, which is the hormone that stimulates red blood cell production, or erythropoiesis, that's happening in the red bone marrow. Okay, so erythropoietin, being a hormone, is going to act like all hormones. It's going to be made in one location and work in a different location. So we already know it's going to work in the red bone marrow, but where it is made is in the kidney. So it gets released in the kidney and it travels to the bone marrow. You're going, well, why is it in the kidney? It's because the kidneys have a sensor system. And what the kidneys are sensing is either you have a low blood count or you have low blood oxygen, because those are the two things that will cause the kidney to release the hormone erythropoietin. So here's how it goes. If you have anemia, the kidney has sensors we will learn about for test number 5. The kidney secretes erythropoietin, which travels in your blood to the bone marrow. It triggers stem cells in your bone marrow to make new red blood cells. And when they are mature, they get kicked out into your blood. And then your blood count will rise back to normal. And you will no longer have your anemia. Now, what if you wanted to climb Mount Everest? Or maybe not Mount Everest, maybe some local mountains, or maybe mountains outside of Seattle. Okay, there's a reason why mountain climbers go and acclimate themselves. They sit at altitude for a week or two before they start climbing. And that is because when you are at altitude, your kidneys recognize, not enough oxygen. And so it kicks out erythropoietin, which has been abbreviated as EPO, which once again stimulates those stem cells in your bone marrow to have those red blood cell precursors make red blood cells. Lots of steps. We're not learning it in this class. Once your red blood cell count goes high, negative feedback on erythropoietin, just like we would have had over here, negative feedback. But now you have more red blood cells. So in this case, you have a more oxygen carrying capacity for climbing at altitude. All right, that's it for this section. Come on back as we talk about some red blood cell related disorders, some fascinating stuff I'm sure you're going to be interested in. See you shortly.